See if I could find my PowerPoint somewhere on the screen. The screen is going blank. Let's see here. I'm looking for my PowerPoint. The screen went blank and now it's shifted location. So uh, give me a moment. Okay. All right. I think I have it. I hope it's the right thing. So today we have the review for urinary system only, which is what I had taught. I'm going to put it on record for those that want it recorded, which is most people. If you have questions, ask them. So let's go ahead. Let me see if I could share. Let me find the record button also. Are you able to see my PowerPoint?
Yes.
Okay, I'm going to press record now.
Recording in progress.
Okay, this should be recording right now. Let me put it on the full screen. Okay, do you see the full screen presenter mode, I guess? Do you see that?
Yes.
Okay, all right. So we could go ahead then. I believe this is recording. It should be recording. Okay, so today we're talking about urinary system. Let's do it. Let's do an overview first. The best way to approach kidney is to see a gross specimen of kidney, or at least a picture of it, and cut it sagitally like we're seeing here. You see cortex and medulla, the outer part. If I draw a line here in this location, you see my pointer, I hope.
Yeah, we can see it.
Okay, so everything peripheral to this is cortex. Everything internal in terms of parenchyma is medulla. So here's cortex, here's medulla. The cortex has these radiations here. These are medullary rays. Internal to the cortex is medulla. Much of the internal parenchyma consists of pyramids. There's roughly 8 to 18 pyramids per kidney. They are somewhat pyramidal shaped on cross section with a flat base here and a pointy apex called the papilla. The between the pyramids are cortical columns like we see here, or renal columns, which also is cortical type tissue that spills over the pyramids because there's so much cortical tissue, it spills over the pyramids, but it is still called medulla. Those renal columns are called medulla, and the medullary rays, which you see in cortex, are still part of the cortex. So that's a bit confusing, but it's something that's not that difficult to remember, I think. The kidney is a bean-shaped structure with a hilus that's rather concave in its appearance. And entering and exiting from the hilus are several structures, including the renal pelvis, which collects urine to discharge into the ureter, and the renal arteries and renal veins. So those are structures we see in the hilus of the kidney. Each papillae of the medulla discharges into a minor calyx. Several minor calyces join together to form a major calyx. Then the major calyces join together to form the renal pelvis, which is funnel shaped that narrows down to form a ureter. The pilocalyceal system is lined by urothelium or transitional epithelium, which we will see more of later, but it is a type of epithelium specialized to be able to be stretched by distension from urine. And it also is resistant to the adverse effects of urine. So if the urine is hypertonic, which it often is, it's able to resist that hypertonicity, protect the body from the hypertonic urine, and able to protect the body from whatever toxins or waste products may be present in the urine. Okay. The basic functional unit of a kidney is a nephron. Here we see a diagrammatic image of the nephron. A nephron consists of this spherical structure called a renal corpuscle or glomerulus, in a sense. So the renal corpuscle or glomerulus consists of a glomerular capillary tuft, a tuft of capillaries, which I'm indicating right here, and a bilayered epithelial capsule, which surrounds that tuft of capillaries. That's together the so-called renal corpuscle or glomerulus. We see the glomerular tuft of capillaries arises from an afferent arteriole, which breaks up into that capillary plexus. And then the capillaries collect together to form an efferent arteriole, which we see illustrated here. That efferent arteriole carries on further and gives rise to another capillary bed, which we'll talk about shortly. Attached and continuous with the epithelial capsule of the glomerulus is a renal tubule. Here you see a renal tubule. It has a number of sections. There's a proximal portion, a proximal tubule. There's an intermediate portion, which we usually call the loop of Henle, and there's a distal portion, which we call the distal tubule. All these parts have several subsections, but let me say that the loop of Henle, anatomically speaking, consists of four parts: a proximal straight tubule, often just called a thick descending limb, a thin descending limb. There's a hairpin turn here, a thin ascending limb, and a thick ascending limb. The loop of Henle is important for creating a concentration gradient within the renal medulla, which is important for absorbing water back into the kidney and creating a concentrated urine. That's more physiology. We won't be talking too much about it today, but it's important to be aware of that. Let's move forward. The blood supply to the kidney is important to be familiar with. It has a very distinct type of pattern because the kidney is a blood filtering organ, so it makes sense to have a particular structure to the vasculature of the kidney. There's a renal artery, which divides into segmental arteries, which further divide into lobar arteries. The lobar arteries skirt the lateral sides of the pyramids, like you see here. These are the lobar arteries. They're actually small arteries. They're something that are very difficult to see in a gross specimen, although you might see them if you look carefully. The lobar arteries travel to the top of the renal pyramids, like you see here, and form an arciform branch over the top of the pyramid. This is called the arcuate artery. It forms an arch over the base of the kidney, like you see here and here and here. And then radially arising from the arcuate artery are these branches, which we usually call interlobular arteries. These are small arteries, very narrow and difficult to see grossly, although you might see tiny bits of these branches grossly. These small vessels are something that is easier to see on microscopic examination. So it's important to know that they are there conceptually, whether you see them in a gross specimen or not, but they are there microscopically, and certainly they are important in terms of understanding how the kidney functions. So these interlobular arteries extend from the base of the renal pyramids towards the capsule of the pyramids. I didn't mention before, but there is a capsule to the kidney. These are the interlobular arteries. Why are they important? Well, one reason they're important is that they give rise to the afferent arterioles, like this you see here. There may be several afferent arterioles coming off of one interlobular artery, and the afferent arterioles then give rise to the glomerular tuft of capillaries, which I mentioned to you earlier. So in the figure on the right, you see again a drawing of several different nephrons and their association with the arcuate artery, the interlobular artery, and the immediate feeder of the glomerular capillary tuft, which is the afferent arteriole. There's also a short segment here of an artery you see coming off. That's the efferent arteriole. Now, when we look at the nephrons in this figure here, there are actually two major classes of nephrons. One is the more superficially located nephrons, which are often called superficial nephrons, or they may be called cortical nephrons. because they tend to be rather high up in the cortex. That you see here on the left-hand side of this box right here. On the other side of that box, the right-hand side of the box, you see a nephron which is closer to the cortex medulla junction. These are called juxtamedullary nephrons. So two major categories of nephrons, cortical nephrons and juxtamedullary nephrons. The efferent arteriole of the glomeruli giving rise to these tubules here actually gives rise to the capillary plexus supplying these different classes of nephrons. So the efferent arteriole of the more superficially located cortical nephrons gives rise to a plexus of capillaries around these more superficially located nephrons. These are the this is the peritubular capillary plexus. It's called the peritubular capillary plexus. The efferent arteriole of the juxtamedullary nephron here gives rise to a capillary plexus, which dives down into the medulla and forms these parallel descending and ascending limbs. This capillary plexus arising from the efferent arteriole of the juxtamedullary nephrons is the vasa recta. These are the vasa recta. So that's a capillary plexus too, essentially. So peritubular capillary plexus arises in the more superficially located cortical nephrons. The vasa recta capillary plexus arises from the efferent arteriole of the juxtamedullary nephrons. Now, several other things to know about these two classes of nephrons. Around 85% of the nephrons in the cortex are the cortical nephrons, or more superficially located nephrons. About 15% of the nephrons in the cortex are the juxtamedullary nephrons. So only a minority of nephrons are juxtamedullary, which give rise to this vasa recta type plexus diving down into the medulla of the medullary pyramids. The nephron loops arising from these cortical or superficial nephrons is short-limbed. You see, the descending and ascending limbs of the nephron loop for this cortical nephron here are rather short. They only dive down into the more superficial part of the medullary pyramid, whereas the nephron loops arising from these juxtamedullary nephrons dive down deep into the medullary pyramids. These are long loop nephrons here on the right-hand side. So that is another difference between the superficial cortical nephrons and the juxtamedullary nephrons. It is the juxtamedullary nephrons with their long loops of Henle, which are the main contributors to generation of the hypertonic gradient of the medulla. Another thing to know is that the vasa recta form loops that are parallel to the loops of Henle of these long looped juxtamedullary nephrons. So you see here the vasa recta, those are rather parallel to the loop of Henle arising from the juxtamedullary nephrons that dive down deep into the medulla. Okay, so let's go to the next picture here. Now we're talking about renal cortex. If you look at the figure on the left, it shows a panoramic view of the kidney histology. This is actually a drawing, so it's very crisp in its appearance, but you see cortex here on top and medulla here. This is a pyramid on the bottom. The cortex and the medulla are clearly different. They are clearly highly organized, but different. The cortex consists of alternating areas containing glomeruli, like you see here. These balls are the glomeruli. And those glomeruli are surrounded by convoluted tubules. This area here, consisting of glomeruli and surrounding convoluted tubules, is the cortical labyrinth. So here's a cortical labyrinth, here's a cortical labyrinth, here's a cortical labyrinth. Now, between the cortical labyrinth are areas with straight, parallel, aligned tubules. These are the medullary rays. So really, the cortex consists of alternating cortical labyrinth, like we see here, containing the glomeruli and convolute tubules, and medullary arrays. So labyrinth, medullary array, labyrinth, medullary array, labyrinth, medullary array, something like that. If you were to look in cortex at higher magnification, you would know what to expect in the cortex. By finding cortical labyrinth and finding medullary arrays, you would know what tubules you would encounter. If you look carefully at medullary arrays at high magnification, you would see collecting ducts, and you would see straight portions of the proximal and distal tubules. In the cortical labyrinth, you have convoluted portions of the proximal and distal tubules. In the medullary arrays, you would see straight portions of the proximal and distal tubules, as well as collecting ducts. Those things are reflected in the drawing on the right-hand side of this slide here. If you look at the cortical labyrinth here, and also here, you see glomeruli, and you see these convoluted tubules arising from the glomerulus, which is the proximal convoluted tubule right here. And if you look around, you also see portions of the distal convoluted tubule as well. So glomeruli and convoluted tubules are the inhabitants of the cortical labyrinth. In the medullary array, you see a collecting duct right here, and you see portions of the straight limbs of the proximal tubule and distal tubule in the medullary array. Now, multiple nephrons converge to form one collecting duct. Here's a collecting duct, and you see multiple nephrons converging together and joining together to form the collecting duct in each medullary array. In the medulla, if you look down here, we haven't yet looked much at medulla in the figure on the left-hand side yet, but you see continuation of the collecting duct in the medulla, and you see fairly straight, aligned, parallel portions of tubules, which constitute much of the limb of the loop of Henle right here. So medulla consists of the loop of Henle as well as the medullary portions of the collecting duct. If you look at the figure here on the left-hand side, you see lots of aligned, straight, parallel tubules consisting of the loop of Henle and collecting ducts. You can't easily make out the distinction between those different types of tubules at this intermediate magnification. But when you look at higher magnification, then you can see the distinction between these various tubules much better. This is a section through cortex. I know it's cortex because I see several glomeruli. I know glomeruli are found in cortex. If I look very close, I see E here consists of glomeruli and convoluted tubules around it. If I look at left, to the left of that at F, I see straight aligned tubules. So this must be a medullary array containing collecting ducts and straight portions of the tubules. Let's look at renal medulla. We talked about what structures you might see in renal medulla. Again, I emphasize the fact that the collecting duct continues onward into the medulla, and here you see portions of the loop of Henle, which are also straight and aligned, and that's what's causing this appearance that we see here on the left-hand side. Now, if you take a cross-section through the medulla, this is the best way to really see the different variety of tubular structures that you may encounter in the medulla. We see a variety of different tubules. We'll talk a little bit more about this later, so I don't want to belabor the point here right now, but there are a variety of different tubular structures in the medulla, including collecting ducts and portions of the loop of Henle, as well as capillaries that belong to the vasa recta. How do I easily recognize a capillary versus one of these renal tubules? Well, capillaries often contain blood. So those structures that look like this with very thin wall and blood in their lumen are most likely the capillaries. The vasa recta and the rest of these tubular structures would be the various types of renal tubules. We'll talk more about this later as we go through this review. Renal glomeruli or corpuscles. Here's a diagrammatic view of those. It's important to have a roadmap to know what you're looking at. It is very hard to really make sense of these complex structures. Just looking at a histologic image, it is much easier if you develop a roadmap in your head as to what you can expect to see and what you are actually seeing in a histologic section. So here's a picture of a renal glomerulus or corpuscle. Here's a ball of capillaries. Actually, this is a whole bunch of capillary loops which are anastomosing with each other. That forms the glomerular capillary tuft. That's what we're seeing here. As we can see by looking more carefully, the glomerular capillary tuft arises from an afferent arteriole. It's labeled here, and it collects together at the very end into a efferent arteriole, which we're seeing here. So those are important associations with the glomerular capillary tuft. The glomerular capillary tuft is surrounded by a bilayered epithelial capsule. The outer layer of the epithelial capsule consists of flattened epithelial cells called parietal epithelial cells. The inner layer of the epithelial capsule consists of these unusual looking, highly specialized epithelial cells, which you might think of as being vaguely stellate or octopus-like in their configuration, but they actually embrace the outer part of the capillary wall very tightly and therefore form part of the capillary wall. And they have these processes wrapping around the capillary wall here. These special visceral epithelial cells, which form part of the capillary wall, which embrace the outside and become incorporated into the outside of the capillary wall, are called visceral epithelial cells or podocytes. Podocytes. And they have processes like you see here at higher magnification. The largest processes are the primary podocyte processes, and those further divide into secondary processes like you see here. The secondary processes are sometimes called pedicels or foot processes, and the pedicels or foot processes interdigitate. They interdigitate like this, leaving a thin slit space between them. So the podocytes have these secondary processes that interdigitate and form slit spaces between them. Another important thing to take away from these images is that the bilayered epithelial capsule, Bowman's capsule, has a space within it. This space between those two layers is called Bowman space or urinary space. This is where the urinary filtrate collects as the blood is filtered in the glomerulus from the liquid urinary filtrate, collects in Bowman's space or urinary space, and then is carried into the proximal convoluted tubule, the first part of the renal tubular system. So here's the piece of proximal convoluted tubule, and you see there's a very distinct microvillus border at the apical surface of the epithelial cells of the proximal convoluted tubule. Because of the great numbers of microvilli, the proximal convoluted tubule epithelial cells actually have a so-called brush border, which we saw earlier in the course in the small intestine. The small intestine has a brush border for absorption of nutrients and so on from the GI tract, from the small intestine. Here we see a brush border because there's absorption of materials from the urinary filtrate starting in the proximal convoluted tubule, and much of it occurs, much of this absorption, or we might say reabsorption, because originally the materials were found within the bloodstream, within the blood, which was filtered at the glomerulus. So these materials are reabsorbed in great amount in the proximal convoluted tubule, as evidenced by the brush border. Now there's a seemingly a triangular or conical gap between the afferent arteriole and efferent arteriole here. At this gap, we have several important structures. We have the distal straight tubule or thick ascending limb of the loop of Henle, as it's also known, comes back to the mother glomerulus, to the parent glomerulus. So this glomerulus ultimately had given rise to a thick ascending limb of the loop of Henle, and here it is. It's come back to this gap between the afferent and efferent limbs of the loop of Henle. Together with several other structures, these make up the juxtaglomerular apparatus. So cells within the thick ascending limb, called the macula densa, are part of the juxtaglomerular apparatus. Several other things are part of the juxtaglomerular apparatus. The macula densa, number one. Within the wall of the afferent arteriole are smooth muscle cells, which are secretory and make the protease renin. So there are renin producing and storing juxtaglomerular cells within the afferent arteriole, present within the afferent right here. Those are another part of the juxtaglomerular apparatus. And thirdly, there are cells in the interstitium between these two arterioles, the afferent and efferent arteriole, and the thick ascending limb here, with its macula densa, this cluster of cells between those structures, are our extraglomerular mesangial cells, also known as Lacis cells, LACIS. So those three elements together are part of the juxtaglomerular apparatus, which are important in regulating blood pressure. And we'll talk more about that as we move on, but we're seeing it here for the first time. And one thing I haven't mentioned yet is that the juxtaglomerular apparatus is occurring at the vascular pole of the glomerulus. Every glomerulus has a vascular pole and a tubular or urinary pole. So this here is the vascular pole of a glomerulus. This here is the tubular or urinary pole of the glomerulus. The vascular pole is where the blood vessels go in and out of the glomerulus, where they give rise to the capillary tuft of the glomerulus and exit from the capillary tuft of the glomerulus. And the tubular pole or urinary pole is where the urinary filtrate is collected and enters into the tubular part of the nephron. So these diagrams give you quite a bit of important information, which give you a roadmap to understand what we're looking at on histology. Here are several glomeruli. As you see, the glomeruli are rather spherical structures, which is what we expect. When we looked at that drawing in the prior slide, we saw distinct capillary loops covered by podocytes, which are rather octopus-like in their appearance. And here, you can't really see that, but if you have a roadmap in your head, you can conceptualize the actual structure of the glomerulus much better. Looking at this glomerulus here on the left-hand side, I see there is a pole present. It is a tubular pole where the proximal convolute tubule is present. Sometimes this is called the urinary pole because this is where the urine first collects in the tubular system. I do not see a vascular pole on the left-hand side. So whether you see a tubular pole or a vascular pole or both together at the same time depends where you cut a glomerulus in your histologic section. If you cut it through the pole, you will see it. If you cut it towards the edges of the glomerulus, you will not see those poles. So it's a hit or miss type of phenomena. In this particular case, we see the tubular pole. Also, you can see quite readily how the urine may collect in this urinary space and then flow into the tubular pole here, where it exits into the proximal convolute tubule, which is right here. As you can see, the proximal convolute tubule is composed of rather cuboidal-looking cells. I presume there's a brush border here. It is often difficult to see the brush border because it's not always super well preserved, but conceptually there is a brush border there, which is involved in resorbing a large number of materials into the via the proximal convolute tubule back into the body. If we look at the glomerulus per se, it is hard to see the different elements of the glomerulus unless you know what you're looking for and have the right kind of preparations. But I presume there will be capillary loops in there, which are lined by endothelial cells. I presume there will be podocytes in there, which form the outer cell type in the capillary wall. And I presume there will be a support type cell, the mesangial cells. And we'll see a little bit later how we can recognize those cells better. On the right-hand side, here's a figure. Again, I see that spherical structure, which is a glomerulus. I know this must be within the cortex of the kidney. I do see a pole here. This is a vascular pole because I see a blood-containing vessel either going in or out. I don't know if it's afferent or efferent. I'm not that concerned about it, but I certainly do a vascular pole to the kidney. I know the juxtaglomerular apparatus must be somewhere nearby. I may or may not be able to see all the elements of the juxtaglomerular apparatus in a given tissue section. But again, I'm not that worried about it. I see whatever I'm able to see, and I don't see what I don't see, and that's it. I don't worry about it further. We'll talk more about these things as we go on. In the background, I know in the cortex, the glomeruli will be surrounded by convoluted tubules. So there's a bunch of tubule transections in the background, which must be the convoluted tubules, either proximal or distal tubules. If we look at this image here, I see two poles to this glomerulus. I see a tubular pole here where I see cuboidal epithelium of the proximal convoluted tubule. I see a lumen of the proximal convoluted tubule. On the other side, I see what looks like a thin-walled blood vessel consistent with an arteriole, maybe afferent arteriole, maybe efferent arteriole, I'm not sure which, with a thin wall and a flattened type of epithelium lining it, which I presume must be the endothelium. So here I'm seeing two poles to this particular glomerulus because of luck, just serendipity. It's just how the glomerulus was cut. Notice, by the way, that the cuboidal epithelial cells of the proximal convoluted tubule are continuous with these flattened epithelial cells, which are part of the parietal layer of Bowman's capsule. So the cuboidal cells transition rather abruptly to the flattened epithelial cells, the squamous epithelial cells of the parietal layer of Bowman's capsule. The epithelial cells which are actually incorporated into this glomerular capillary tuft must be the visceral epithelial cells, and they're quite challenging to see, but if you know what to look for, you can find them. We'll talk more about that. Here's another glomerulus. It's a nice section, perhaps even a better section than the one we just saw. But here I'm not easily seeing two poles. I know they must be present. They're just not particularly present in this image. But I could make out various structures. I could see a glomerular tuft of capillaries, which I know contains a variety of cell types, the podocytes, the endothelial cells, and the mesangial cells. I also know that the capillary loops are surrounded by or contain glomerular basement membranes as part of the capillary loop. And if I look carefully, I see what looks like a line you see here, like a loopy looking line, like right there and right there and right there and right there and right there. You see that line is actually, that loop or line is actually the glomerular basement membrane. So you can make out glomerular basement membranes by looking at a cross section of a glomerulus. There are easier ways to see it. If you do special stains, you could see it better, but I can even see it on light microscopy. As I said to you earlier, the outer layer of the epithelial capsule consists of flattened epithelial cells, the parietal epithelial cells. They're squamous, if you want to call them that. Here they are. And then here's a capillary loop. There's a lumen. That's how I know it's a capillary loop. And that lumen is surrounded by this line, which I think is glomerular basement membrane. And then outside the glomerular basement membrane are these flattened cells, which are podocytes. The podocytes are on the outer part of the capillary loop, outside the glomerular basement membrane, if you will. These are podocytes. I cannot see the interdigitating processes easily here in this kind of image, but I know they are there. They are types of things you would see more easily on electron microscopy. And that's one reason people do electron microscopy and kidney biopsies, because you can't see all the details you would like to see to evaluate glomerular disease. If a nucleus appears facing the lumen, even bulging into the lumen like we're seeing here in this image, we can judge that that must be an endothelial cell. So right now, I showed you how we could recognize an endothelial cell and how we could recognize a podocyte. Now, I'm not seeing the audience here in my particular image, but I think people have been trying to maybe get my attention. Let me see if I could. I don't want to mess with this presentation too much, but I'm
Yes,
not
Dr. Brown, I had a
seeing
question real
the
quick.
gallery. In other words, so if you have a question in my particular presentation on my screen, so if you have an image, if you have a question, maybe speak up. That way I know you're trying to ask me a question.
Okay, Dr. Brown, I have a question. So you were pointing out the basement membrane in this glomerular capsule. Is it safe to say that it's also viewed as the visceral layer? Because you said the parietal layer was on the outside and then it envelopes for the visceral. Is that the same in these images or no?
Yes.
Okay.
So parietal layer, those flattened cells on the lining Bowman's capsule. These out here are the parietal layer. Like parietal
Yes, sir.
pleura is on the outside. And then you
Mm-hmm.
have a space, the urinary space here, Bowman's space, which is collecting the filtrate. And then on the glomerulus itself, the tuft of capillaries itself, you have epithelial cells. Usually it's hard to recognize them, but here you can recognize them because they're on the very surface of the glomerulus, and these are outside the capillary lumen. They're outside the lumen. They seem to be on the other side, right?
Mm-hmm.
The
Mm-hmm.
capillary lumen. So these are podocytes or visceral epithelial cells, same thing. Podocytes
Okay.
are the same thing as visceral epithelial cells. They're inside the urinary space. See, this is the urinary space. They're inside, and they sit really as part of the glomerulus itself, the glomerular tuft of capillaries itself.
Thank
Okay.
you.
So again, I'm not seeing the gallery and whether people are raising their hand or not on my presentation. I just don't want to play with the screen too much. I'm afraid I'm going to lose something. So if you have a question, just speak up and I'll try to answer it. So here's the next picture. And I said to you, you could see the glomerular based membrane and other features of the glomerulus on H&E. And H&E is very useful, but you see them better with special stains. And here's an example of several special stains. PAS is often done, and it will show you the glomerular-based membranes quite nicely. You see they stand out a little bit more because there's glycoprotein constituents within the glomerular-based membranes, and within the mesangial matrix as well. So those structures will stand out better on a PAS stain. Now, we don't need to know too much about that, but in real life, when people do kidney biopsies for kidney diseases, including glomerular diseases, pathologists will do special stains to get a better look at the morphology of the glomerulus. So that's why it's important to know a little bit about them. If you compare this picture of PAS with the H&E, you see some details which you cannot see easily on H&E. Another special stain, which is commonly done, might be the Jones methenamine silver stain. It's a silver stain and the basement membranes and the mesangial matrix stained black, like you're seeing here in this image. So again, it shows you some features of glomerular histology which are not easily visible on H&E, and they are done in real life, although we don't have to worry that much about that right now. You worry more about this when you study pathology. One really good way to see the structure of glomerulus on histology, even better than you see in some ways than on a paraffin section, is to do a plastic section. So a plastic section is cut thinner. It's fixed better in some ways than a typical formaldehyde fixed paraffin section. It's cut thinner because plastic is hard and easier to cut at a thin level. And it also could be used to prepare for doing electron microscopy, which for renal pathology is a very important procedure. Doing electron microscopy is not an academic exercise for us to study histology and understand it. It's actually part of the way actual kidney biopsies are done in real life. So here's a plastic section of a glomerulus, and you could see details which are not visible easily otherwise. So let's go through some of these details. Here we see a glomerular tuft of capillaries. This, by the way, is an arteriole, either an afferent or efferent arteriole. I don't know which, entering into the hilus, in a sense, of a glomerulus at the vascular pole of a glomerulus. Here we see a bunch of capillary loops. You see how the empty space here would be where the blood is during life. The blood has been drained out, but there would be blood within these spaces and these capillary loops. You see here what looks like a blue line around that space. That's the glomerular basement membrane. And then there's a variety of cell types. So again, Here is Bowman's space where the urinary filtrate is collected on the outside of that space, on the outside of Bowman's capsule, and lining the outside of Bowman's capsule are the parietal epithelial cells. They are flat epithelial cells, like you see here. These flat epithelial cells lining the outside of Bowman's capsule are the parietal epithelial cells. Parietal epithelial cells. They are squamous in configuration, if you want to call it that. They are flattened epithelial cells. Because they're flattened, it's easy to forget that they are epithelial cells, but they are epithelial cells. The podocytes, the visceral epithelial cells, are found on the outside of the capillary loops. There's a podocyte right there, and there's a podocyte right there. You see? That's on the inside of Bowman's capsule, on the outside of the capillary loop. Let's look at the next picture. There, an arrow is pointing towards a visceral epithelial cell or podocyte. You see there? A visceral epithelial cell or podocyte. There, a visceral epithelial cell or podocyte. It's the inside of Bowman's capsule. It has that octopus-like look. If you look at a figure, a drawing of the structure of glomerulus, and if you look on electron microscopy, then you will see the actual configuration of podocyte even better than this. But this is pretty good. This urinary space is where the filtrate is collected. Okay. Here's another arrow pointing towards a capillary loop. We see a nucleus with a thin rim of cytoplasm bulging into the lumen of that capillary loop. That's a endothelial cell, an endothelial cell, because it is bulging into the lumen of the capillary. And actually, if you look very carefully, the endothelial cell seems to be on the outside of the glomerular basement membrane. This line here, being glomerular basement membrane, the endothelial cell is on the inside of that. So the podocyte is on the outside of the glomerular basement membrane. The endothelial cell is on the inside of the glomerular basement membrane. So on a plastic section, you could see quite nicely some of these structures that are normal parts of a glomerular capillary tuft, and you see their relationship even better than on H&E. And this is what would actually be done in a real kidney biopsy for glomerular disease. Here's another arrow pointing towards a cell which seems to be in the interstitial space between capillary loops. So here's a capillary loop, here's a capillary loop, here's a capillary loop, here's a capillary loop. Even here, perhaps, you see this kind of a ring of capillary loops, and there's an interstitium space, an inner between the capillary loops. Now, between capillary loops, they're being supported by a stalk of interstitial tissue. The stalk of interstitial tissue forming a pedestal or support for the capillary loops is the mesangium. And the mesangium consists of cells known as mesangial cells and extracellular matrix known as mesangial matrix. So here we see that stalk-like interstitial support for several capillary loops, which is the mesangium. So this here is mesangium, and this arrow is pointing towards a mesangial cell. And the extracellular matrix here, which is a little bit darker, perhaps, I'm not sure that's true, but darker perhaps than the glomerular based membrane, is the mesangial matrix. Mesangial matrix. So here's a mesangial cell and surrounding mesangial matrix of a glomerulus. So I just showed you a way to identify different components of a glomerulus. Now, don't worry if you cannot identify every single element in a given section of glomerulus. I mean, I cannot necessarily identify every single nucleus, every single element of a glomerulus in a tissue section, but I could identify some of them at least if they're typical. I could identify some of them, and that's all we really want to do. So don't worry, don't think that you necessarily have to identify every single cell that you find in a glomerulus. Here's a higher power view again of a plastic section of glomerulus, and you see here a ring of capillary loops like this, a ring of capillary loops. There are some red blood cells in the lumen of that capillary right there. The capillary loops are demarcated by a glomerular basement membrane. That dark line forming a loop is a glomerular basement membrane. I know that on the inside of a glomerular capillary loop is going to be an endothelial cell. So this cell right there is labeled, by the way, here on the side, is an endothelial cell. it bulges into the lumen. Here's another endothelial cell right here. If you look very carefully, I could see that the endothelial cell is actually internal to the glomerular basement membrane. I know also there will be podocytes on the outside of the glomerular base membrane. So if we look down here, there's two cells with their nuclei and a bit of cytoplasm around them, which are podocytes or visceral epithelial cells. So here are podocytes or visceral epithelial cells. They are also labeled in the diagram here. So again, I've been able to identify, if I look carefully and know what to look for, endothelial cells, podocytes. Now, I know capillary loops seem to be supported by something of a pedestal, if you will, or between the capillary loops is this interstitium supporting them, which is called mesangium. So here I see the interstitium supporting the capillary loops. The cells within this interstitium are therefore the mesangial cells in the extracellular matrix in which the mesangial cells are embedded is the mesangial matrix. Now, mesangial cells have several important functions. They are contractile. They may help control the diameter of some of these capillary loops. They are phagocytic. They may help remove protein aggregates and other materials which may get caught in the glomerular capillary filter. The capillary wall, I don't think I necessarily mentioned this yet, but the glomerular capillary wall actually is the glomerular urinary filter. That is the filter. So when the blood is pushed from the capillary lumen into the urinary space is the glomerular capillary wall in its entirety, which it makes up the glomerular filter. So here we have the mesangium forming the pedestal supporting several capillary loops. We could see all these things again on electron microscopy and even in more detail. Here we see several capillary loops, which are being supported by a central pedestal or core of mesangium here, containing a mesangial cell and mesangial matrix. Each capillary loop has a wall consisting of three major elements. On the inside of the capillary wall is the endothelial cell. In the middle of the capillary wall is a layer, which is the glomerular basement membrane. Here's the glomerular basement membrane. On the outside of the capillary wall is the visceral epithelial cell or podocyte, forming primary processes and then pedestals. So these are all, these little nubbins here are pedestals coming off of the podocytes. All together, the endothelial cell, the glomerular basement membrane, and these pedestals of the podocytes form the glomerular capillary wall, which is the glomerular filtration barrier. That capillary wall in the glomerulus is the glomerular filtration barrier, consisting of the three elements I just told you about. If you look at the electron micrograph, I see something very similar. I see several capillary loops. Here's a capillary loop here. Here's a capillary loop here. Supporting them is mesangium with a mesangial cell, which is and mesangial matrix, which structurally supports the capillary loops of the glomerulus, but they also have other activities like their ability to phagocytize protein aggregates or get stuck in the glomerular filter. They may be contractile, and they may have several other functions as well. And these are the glomerular capillary loops, which must have three layers to them. If we look carefully, we should be able to find three layers of them, and I will go over them in a moment. Here's a scanning electron micrograph, which we could compare to a transmission electron micrograph here on the right-hand side. You see in the scanning electron micrograph where the capillary loops are twisting and turning in a three-dimensional appearance, we have on the outside of the glomerular capillary a cell with a body, that's the podocyte. The body has major processes coming off it, which are the primary processes. Coming off those primary processes are smaller foot processes or pedestals. We see the pedestals are interdigitating like this. They like you take your hands with two fingers and interdigitate the fingers together, and between those fingers is a slit space through which the filtrate has to push, right? That slit space is very narrow and prevents materials which should not be filtered into the urinary filtrate from going into the urinary filtrate. It keeps them in the bloodstream, in a sense. So the characteristics of the pedestals and their filtration slits is very important in maintaining a normal urinary filtration barrier. If we look on the right-hand side, we see a capillary loop right here. We see a cell which seems to be sitting just external to the capillary loop right here. This cell is a podocyte or visceral epithelial cell. The podocyte or visceral epithelial cell has a big, thick process like right here and right here. These are primary processes. Those give rise to secondary processes, which are the foot processes or pedestals. And you see here how the pedestals or foot processes adhere to the external aspect of the glomerular capillary wall. If you look very closely, you see the inside of the glomerular capillary wall as a very thin cytoplasm, which is the endothelial cell cytoplasm. It's quite interrupted because there are lots of fenestrations. The endothelium of the of the glomerular capillaries is fenestrated, has openings, fenestrations or pores, fenestrations. And we can see that here in the endothelial layer. The middle layer consists of this grayish line, which is the glomerular basement membrane. And then on the outside of the glomerular capillary wall, you see the pedestals of foot processes being elaborated by podocytes. So even at this magnification, we cross-section, in transmission electron microscopy, we can see 3 layers to the glomerular filtration barrier. Here's another section showing 2 different magnifications of the glomerular filtration barrier, which, as I said to you before, is actually the capillary wall, the glomerular capillary wall. The inside of the glomerular capillary wall consists of the fenestrated endothelium, which is quite flat. The middle layer, you have to look carefully because it's hard to see, is the glomerular basement membrane. And the outside of the glomerular capillary wall are the pedestals, right here. The pedestals, okay? 3 layers to the glomerular capillary wall, the glomerular filtration barrier. If we look at even higher magnification, we see here's the lumen, the capillary lumen, where there would be blood. Here's the urinary space where there would be a urinary filtrate. By the way, which parallels what we see here in the left-hand figure. I didn't mention this. Here's blood in the lumen of the capillary. That's this space. This space here is the urinary space containing the urinary filtrate. Well, going back to this higher magnification view, we see the flattened endothelium of the capillary, the fenestrated endothelium. It has fenestrations like right here, for example. In the middle layer, we see the glomerular basement membrane. This is the glomerular basement membrane. And the outer layer here, facing the urinary space, are the pedestals being made by the podocytes. Now, what we see here that we couldn't see earlier is that the slit spaces between the pedestals actually has a interesting diaphragm between them. See here? Here's a glomerular. Here's a slit. You might call a slit diaphragm, bridging this pedestal and this pedestal. Here's a slit diaphragm bridging this pedestal and this pedestal. A slit diaphragm bridging this pedestal and this pedestal. So these slit spaces are bridged by a diaphragm, sometimes called a slit diaphragm, which has several important proteins that make it up. If anything becomes abnormal in the proteins making up the slit diaphragm, the glomerular filtration barrier gets abnormally leaky, and these people develop a variety of different types of glomerular problems. So important to realize that the slit spaces are bridged by slit diaphragms, and they're visible here. The fenestra of the endothelial cells in the kidney are not bridged by a diaphragm. It is the slit spaces elaborated by the pedestals which are bridged by a slit diaphragm. And here it is in this figure. Here's a higher magnification showing you the three elements of the glomerular capillary wall, which make up the glomerular filtration barrier. Here's the lumen of a capillary. You cannot see the blood cells in here, but this is the lumen of a capillary here. On the inside, you have fenestrated endothelium. The openings in the fenestrated endothelium are fenestrations, that's all. They are indicated by arrowheads. Remember, this is an arrowhead. It is not an arrow. The thing between the inside and the outside of the glomerular capillary wall is the basement membrane. It gets a little bit complicated in this image to recognize the glomerular basement membrane because actually there's a dense layer and two pale layers on either side. There's a central dense layer, and then there's a pale layer inside and a pale layer on the outside. All these three layers actually layer parts of the glomerular basement membrane. So don't be confused by the fact that there seems to be a difference in density between these three different layers of the glomerular basement membrane. That's normal. On the outside of the glomerular capillary wall are the pedicels. They look like little feet, if you will. Here's a little foot. These are pedicels. Bridging the pedicels are the slit diaphragms. There's this arrow. It's not an arrowhead. It's an arrow. It's pointing towards a slit diaphragm. Again, another arrow pointing towards a slit diaphragm. The pedicels need to be intact, and the slit diaphragms need to be intact in order for the normal filtration barrier to function properly. They contain a variety of important proteins, which we didn't talk about too much in our course. For example, nephrin. I'm giving you one example of a protein because it sounds like a kidney type protein. It's easy to remember. If something were to go wrong with that protein, a person would develop very leaky glomerular filtration barriers. There would be too much protein appearing in the urine if there was failure of the glomerular filtration barrier. Let's talk about the
For the
Yeah.
Dr. Braun, for the nephrin protein, is that specifically in the basement membrane or is it in the podocytes or the slit diaphragm? Where is the nephrin at?
In the slit diaphragm. I just mentioned to you because it's easy to remember the name nephrin. That's
Of
all.
course. And then for the arrowhead, is that just a fenestrated pore or
Arrowhead is a fenestration. It's
Okay.
fenestrated endothelium. This is a fenestration in the endothelial cell. That's it. The
Thank
arrowhead.
you. Yes, sir.
It's rather flat and horizontal looking, whereas the podocytes or foot processes or pedicels, if you remember foot process, it helps you remember their appearance better. You see, they remind me of little feet because they're vertically oriented and even sometimes have what looks like a foot coming off the leg part of the little feet, right? So these are pedicels of foot processes. This is fenestrated endothelium. This is where you see the diaphragm. This is not where you see the diaphragm of the glomerular filtration barrier. Looking at this again, there are three elements to the glomerular filtration barrier: fenestrated endothelium, glomerular basement membrane, and foot processes or pedicels with their slit diaphragms, with their slit spaces and slit diaphragms. Those are the three main elements of the glomerular filtration barrier. Is that clear?
Thank you.
Yeah. Juxtaglomerular apparatus. This also has three main elements. It's interesting. Juxtaglomerular apparatus has three main elements. The glomerular filtration barrier has three main elements. I mentioned this to you earlier when we first saw a drawing of a glomerulus. So here's another drawing of a glomerulus, and you see two poles to this idealized glomerulus. You see a vascular pole here, and you see a tubular or urinary pole here. I mentioned to you that at the vascular pole, that's where you see the juxtaglomerular apparatus. The juxtaglomerular apparatus consists of three elements. The thick ascending limb of Henle, also called sometimes the distal straight tubule, or even distal convolute tubule. See in this diagram, It's called distal convoluted tubule. Different places have different terminology, so it can be confusing. I want to help prevent some confusion. The juxtaglomerular apparatus, one way to think of it is that it occurs at the end of the loop of Henle, at the junction where the distal straight tubule becomes the distal convoluted tubule. So it's kind of like at that junction, if you will. So some people feel they would rather call it distal convoluted tubule. Some would rather call it distal straight tubule. Some will call it thick ascending limb. All of those are essentially correct. So in any case, at the termination of the loop of Henle, the tubule comes back to its parent glomerulus and inserts itself at the vascular pole between the afferent and efferent arteriole. And that's what you're seeing in this diagram here. The termination of the distal of the loop of Henle, the so-called distal tubule, if you will, is inserting itself between the afferent and efferent arteriole. Now, the nuclei and cells of the thick ascending limb or distal tubule become taller and thinner and more densely packed together, forming a plaque or patch of taller, densely packed cells. This patch or plaque of tall, densely packed cells is called the macula densa. Macula densa. So here's the macula densa of the distal tubule right there at the vascular pole. A second element of the juxtaglomerular apparatus is that nearby in the afferent arteriole, here's the afferent arteriole, some of the smooth muscle cells in the wall of the afferent arteriole become specialized to become secretory cells. So not only are they smooth muscle cells, but they are also secretory at the same time. These are highly specialized smooth muscle cells that are also secretory. They have secretory granules within their cytoplasm, which store the protease renin. They store the protease renin. And thirdly, as part of the juxtaglomerular apparatus, there's an interstitium, which is rather triangular or conical in its shape, between the afferent and efferent arterioles and the macula densa. This triangular space contains cells which are extraglomerular mesangial cells. These extraglomerular mesangial cells are sometimes called Lacis cells, Lacis, French word for lace, because they have processes which somebody thought looked lace-like in their appearance. This network of processes looks like lace. So those are a third element. The Lacis cells are the third element of the juxtaglomerular apparatus. What is the purpose of a juxtaglomerular apparatus? Well, I guess simplistically speaking, it helps in regulating total body fluid balance and blood pressure. Fluid balance and blood pressure are closely related. You need to retain fluid, salt, and water to maintain fluid balance, to maintain your blood pressure. So the macula densa is assessing within the fluid within it the salt concentration of sodium chloride, and the fluid flow within the lumen. So the macula densa is a sensory structure sensing fluid flow and salt content. And if it gets to be low, it helps your body raise its blood pressure by communicating with the juxtaglomerular cells within the afferent arteriole. See, they're very close by. They're nearby. It sends a paracrine message to the juxtaglomerular cells of the afferent arteriole to release the protease renin. The protease renin starts off the renin angiotensin aldosterone system, which causes the body to retain more salt and more water, causes the body, especially the kidneys, to retain more salt and more water to help raise body fluid and body blood pressure, body fluid, body blood pressure. So this helps regulate those things. It's very important maintaining our blood pressure, especially if you lose blood pressure, if you lose fluid, if you lose blood pressure. The juxtaglomerular apparatus, which is monitoring every single glomerulus in a sense, is helping ultimately to raise the blood pressure. Now, on the right-hand side, you see a cross-section of glomerulus with the vascular pole, and a tubular pole, and you see the elements of the juxtaglomerular apparatus at the vascular pole in this figure. Here in this figure, you see even better, I think, the fact that the extraglomerular mesangial cells are in continuity with the intraglomerular mesangial cells. You see here, the intraglomerular mesangial cells form this stalk-like um, core or center supporting and helping structure the capillary loops of a glomerulus. Well, the intraglomerular mesangium is continuous with the extraglomerular mesangial cells, which we see here. As I said to you earlier, another name for the extraglomerular mesangial cells is the Lassie cells. These are Lassie cells, which are just another name for extraglomerular mesangial cells. And you see that better in this particular figure on the right-hand side. Another thing I think you see well in the figure on the right-hand side, you see the juxtaglomerular cells, which, despite their name, are actually modified smooth muscle cells and make the protease renin, which is responsible for starting off the renin, angiotensin, aldosterone system, helping maintain body salt and water content and blood pressure. Is that clear?
Yes.
Okay. So how do you translate that figure into an actual histologic section? Here's an actual histologic section taken originally from our textbook. And number one, I see it is a vascular pole. It's part of a glomerulus. You're not seeing the entire glomerulus, but I could still make out elements of a glomerulus. For example, I could see capillary loops quite easily here, right? I could see the urinary space right here, and I could see the parietal layer, Bowman's capsule right here with its flattened parietal epithelial cells. Well, this is a vascular pole. I see here an afferent arteriole labeled right there. I see an efferent arteriole labeled right there. I see what looks like a renal tubule right here. It's a bit collapsed looking or flat looking. I think it's just the way the section came out during processing, but it is a tubule. And I see that part of the tubule closest to the glomerulus actually has taller cells than usual, thinner cells than usual. See, normally they would be rather cuboidal, but here they're even taller than that, and they're more densely packed. They're thinner and more densely packed than usual. This is the macula densa, the macula densa. So I see two arterioles, and I see the macula densa. I presume this tubule came from this glomerulus. This is the mother glomerulus, and it's coming back to the juxtaglomerular apparatus located at the vascular pole of the glomerulus. Then there would be inside the wall of the afferent arteriole, I presume there would be some smooth muscle cells which are modified. They would contain within their cytoplasm secretory granules containing renin. The nuclei of those smooth muscle cells probably would be more rounded. It's hard to see, I think, in this image, but they would probably be more rounded than the usual smooth muscle nucleus, which is more elongated or spindle-like in its configuration. It's hard to see that, but conceptually, I know that would be the case. I'm sorry, I have to just turn my email thing on my just a second. Hello, I'm doing a Zoom session. I'll call back after my Zoom session. Okay, okay, thank you. Okay, that's IT. Nicely calling me doing the Zoom. Okay, so and then the third element, which I haven't mentioned yet in this figure, is that there's this cluster of cells between the arterioles and between the distal tubule, or with its macula densa, that is some of the extraglomerular mesangial cells, or Lassie cells. They're a bit hard to see easily in this image, but you could see them if you know what to look for, and they are labeled L here, L for. So I presume there are some Lassie cells in there that are extraglomerular mesangium. So you could see those things, and if you know that they are present, you know they are there. Those are the elements of the juxtaglomerular apparatus, which I just mentioned to you. Remember, again, that the juxtaglomerular apparatus is important for maintaining your blood pressure. If you lost fluid, for example, or became hypotensive for some reason, there would be decreased fluid flow within the loop of Henle and decreased fluid reaching the macula densa. Also, there may be diminished salt being delivered to the macula densa, decreased sodium, decreased chloride. And there are actually going to be some other factors involved here as well, which we haven't talked about. You may learn about them more in physiology, but this apparatus senses these type of changes and helps the body restore to help absorb more salt and more water, restore fluid content, restore blood pressure via one of the principal ways is by the release of renin from the juxtaglomerular cells, helping to trigger the renin-angiotensin-aldosterone system, retaining more salt and more water, raising blood pressure. Renal tubules. So there are renal tubules in both cortex and medulla. This is cortex. How do I know it's cortex? Well, I see a glomerulus quite easily. I know the glomerulus of the cortex is surrounded by convoluted tubules. So here's a whole bunch of tubules, which I presume are convoluted tubules. I know I can identify, if I look carefully, both proximal convoluted tubules and distal convoluted tubules. Actually, usually you see more proximal convoluted tubules than distal convoluted tubules because the proximal tubule is longer. It's longer than the distal tubule. Most of the absorbing of the kidney, two-thirds of the absorbing of the kidney is occurring in the proximal convoluted tubules. So it absorbs most stuff into the kidney. And therefore you see more profiles of the proximal tubule, usually, okay? The proximal convoluted tubule, being a workhorse of absorption, has cells which are bigger in their volume compared to the distal tubules. They tend to be taller than the distal tubules, and they tend to be pinker than the distal tubules because they contain lots and lots of mitochondria, perhaps even more mitochondria than the distal tubules, although the distal tubules contain lots of mitochondria as well. So looking at this, I see cells which are taller, bigger. The nuclei are more spaced apart because the cells have more cytoplasm and therefore they will be more spaced apart. So these right away look like proximal tubules. In addition, the proximal convoluted tubules have a brush border. Now, the brush border of the kidney sometimes could be a bit challenging to see. It could be a bit raggedy looking. So I take it when I see a raggedy looking apical border to the cell, I take it to be probably the brush border of a proximal convoluted tubule. So looking at these again, I see bigger, taller cells, more widely spaced nuclei, pinker cells, and a raggedy looking border, which probably reflects the brush border. That is the rich a complement of microvilli in the apical surface of these cells. In contrast, there's this cell here, this tubule here, this tubule here, this tubule, this tubule, this, which are distal tubules. These cells have smaller volume. The nuclei tends to come closer together, so the cells look more densely packed. The cells tend to be shorter. The cells tend to be more pale. Not always, but they tend to be more pale, and they tend to have a sharper luminal border. See, the luminal border here is sharper. So these are distal tubules. So I only have two tubule choices here. I have proximal tubules and distal tubules. It's easy, I think, if you pay attention to the details I just mentioned to you, to distinguish between proximal tubules and distal tubules. Here's another picture. Every preparation has a slightly different appearance. So here we have proximal tubules. Proximal. Here's distal. Proximal are bigger cells in volume. The nuclei get pushed farther apart. The luminal border looks a little bit more raggedy looking, presumably reflecting a brush border. If you can compare it to the distal tubules, these are shorter. They're a little bit not as tall as the proximal tubule cells. The nuclei get more densely packed because the cells have less volume. And the brush border is lacking, I believe. There's no really, the apical lumen surface here looks rather sharp compared to that of the proximal tubule. So I made out the difference between proximal tubule and distal tubule. Remember, the proximal tubule does most of the absorbing of the kidney, most of the reabsorbing of the kidney, because originally the fluid came from the blood, so stuff now is being reabsorbed back into the body. The distal tubule also is active. It's metabolically active, doing lots of important things, but less reabsorption compared to the proximal tubule. Here we see the bigger, taller cells, more cytoplasm, more widely spaced nuclei, more pink cells, more raggedy border of the proximal tubules. Here, here, and here, and here. Compared to the distal tubules, here's distal, here's distal, here's distal, here's distal. Now, sometimes if you have the right preparation, you can see a brush border more easily. So here's a funny looking apical surface, which is the brush border in this particular proximal tubule, in this particular preparation. And if you want to see the brush border even better, perhaps you could do a PS stain, as you may recall from earlier in the course. Brush borders tend to have a rather thick glycocalyx, coat. And you could see glycocalyx by doing a PS stain because there's carbohydrate in the glycocalyx. So in this preparation here, the glycocalyx is staining the brush border of proximal convolute tubules. Also, incidentally, the PS is staining basement membrane of these tubules. The renal tubules have basement membranes because they are epithelial cells. We have a basement membrane between the epithelial cells, which are cuboidal roughly, and the interstitium, which is very thin, very narrow in amount, very little interstitium here, but there is an interstitium. Here we see electron micrograph of a proximal convolute tubule cell that has certain specializations characteristic of proximal convolute tubules. There is a brush border. MV stands for microvilli, so there are myriads of microvilli. So many of them that it creates the sense of a brush border, which you see very nicely on electron microscopy, and you see also on a light microscopic specimen if you know what to look for. At the basolateral parts of the cell, like right here, for example, the cell membrane is thrown up into large numbers of basolateral folds to increase the surface area of the membrane so that you could put in ion transporters and other transporters within the basolateral membranes. So there are transmembrane proteins, transporting proteins, among others, inserted into this greatly redundant, enfolded basolateral membrane. It's also typical for proximal convolute tubule cell. In addition, you need lots of energy to drive this transcellular transport of water and solutes. And this is helped by the fact that there's lots and lots of mitochondria making ATP for energy. So there's lots and lots of mitochondria inserted between the folds of these basolateral invaginations. Now, you may wonder how the proximal convolute tubule cell differs from the distal convolute tubule cell. They look somewhat similar on electron microscopy, but what the proximal convolute tubule has that the distal convolute tubule does not have is a brush border. See here, lots and lots of microvilli, myriads of microvilli forming a brush border for the proximal convolute tubule cell, not seen in distal convolute tubule cell, not seen here. This has some microvilli, some stubby microvilli or microrejections, but it is not a true brush border in the distal cell. The distal cell does have basolateral invaginations, does have mitochondria in large amounts, similar to what you see in the proximal cell, but it does not have a brush border. So this is even better. The proximal cells are better, more active even than the distal in terms of reabsorbing stuff, in terms of its metabolic activity. Here we see a cross section through the medulla. You could compare it to this drawing of the medulla here, the diagram showing you what you might expect to find in medulla. If you look carefully at the medulla in this drawing here, you might see, depending where you take a cross section, you might see thick limbs of Henle, you might see thin limbs of Henle, you might see collecting ducts, and also not see in this drawing, you might see vasa recta. So those are the kind of things we may encounter in medulla, and indeed we do encounter them in this particular cross section of medulla. If we look at this figure here, I see an arrow pointing towards the tallest duct here. You see the tallest duct and the palest duct, and the duct with distinct intercellular lateral boundaries like that. All those things are characteristic of the collecting ducts. So collecting ducts have the tallest cells, in this case columnar cells. They have distinct lateral boundaries, like we see here. It's actually the so-called principal cells. The principal cells of the collecting duct which are responsible for this appearance. Those are the cells which are important in reabsorption of water. Under the influence of anti-diuretic hormone, the principal cells of the collecting ducts are insert into their membranes aquaporins, which help absorb water from the from the lumen into the interstitium to be carried back to the body via the capillaries or vasa recta. So reabsorption of water under the influence of anti-diuretic hormone is an important function of the collecting ducts. And you can recognize the collecting ducts here as those ducts with the tallest, palest cells with the nice intercellular boundaries. The vasa recta are also fairly easy to recognize because they're capillaries containing blood. Here's a capillary containing blood. It has a flat endothelium, of course, contains blood. Here's a capillary containing blood. That's vasa recta. Capillary containing blood, 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 blood. Those are all vasa recta. And that leaves the limbs of the loop of Henle. The limbs could be thin limbs. They could be thick limbs. So here's a thin limb of Henle. Oops, let me see. Here, this arrow, the blue arrow is pointing towards a thin limb of Henle. It kind of looks like a capillary with a thin layer of epithelial cells facing the lumen. These are just thin epithelial cells, squamous epithelial cells. But there is no blood in the lumen, no blood in the lumen. Therefore, this is thin limb of Henle, lined by squamous epithelial cells, no blood in the lumen. In contrast, it leaves one more tubule, which we haven't identified yet. This cuboidal epithelium here must be thick limb of Henle because it doesn't fit in any other category. There's no other category it fits in. So here's thick limb of Henle here. The black arrow, thick limb of Henle right there, black arrow. So by careful analysis of picking on the easiest things first, the collecting duct and then the vasa recta capillary, doing those first, those were easiest. Then we had two leftovers, the thin limb and thick limb of Henle. We were able to figure those out quite readily, and that's it. We figured out the medulla. The black arrow here is indicating vasa recta. So loops of Henle in general are important for creating a hypertonic gradient in medulla so that water can then be reabsorbed from the collecting ducts when the collecting ducts are stimulated by anti-diuretic hormone. They normally are impermeable, rather impermeable to water, but under the influence of anti-diuretic hormone, they allow water to flow from the tubule and be reabsorbed back into the interstitium of the kidney and then flow into the vasa recta capillaries and carried back into the body and helping maintain blood pressure. These are these are collecting ducts. They have very distinct lateral cell boundaries, typical for collecting ducts. They are columnar cells, also typical for collecting ducts. Their cytoplasm is often quite pale, typical for the principal cells of collecting ducts. So all the features here, a sharp apical lumen border, by the way, right? The lumen is sharp. They don't have a brush border. Collecting ducts do not have a brush border, so they don't have a ragged or brush border appearing lumen. Another picture of collecting duct, all the same features, except it's a single duct, even without the fact that you have other things to look at. You can tell this is a collecting duct. Columnar cells, sharp lateral boundaries, sharp luminal border, relatively pale cells, typical for the principal cells. These cells are under the influence of anti-diuretic hormone, allow water to be reabsorbed into the hypertonic interstitium of the medulla. Okay, let's talk about the
Dr. Braun,
excretory system of the kidney, the polycal cell system, ureters, bladder, and urethra. Yes.
I'm just having a hard time being able to differentiate the distal convoluted tubule and the collecting duct. Is there a way that you can kind of give a way for us to figure that out?
Well, the distal convoluted tubule is found in cortex. Also, it is not tall. See, the collecting duct can be columnar, the distal convoluted tubule is short. It's shorter even than the proximal convoluted tubule. It's a cuboidal cell. It's not that tall a cell. The collecting duct can be quite a tall cell, number one. So, number one, the distal convoluted tubule is found in cortex, near glomeruli, and near proximal convoluted tubules, whereas the collecting duct, oh, I see. The collecting duct is, okay, maybe I see where your confusion might be. The collecting duct, there's also a cortical collecting duct, and there's a cortical collecting duct and a medullary collecting duct. The cortical collecting duct is found in a medullary array, for one thing, in a medullary array. It is found among other parallel straight appearing tubules. It is not found among the glomeruli and other convoluted tubules. That's one way you distinguish them. The location within cortex. And as I said to you earlier, the collecting duct still looks rather distinct compared to the distal convoluted tubule. It has these very sharp lateral boundaries, which the distal convoluted tubule would not have.
Got it. Thank you.
Okay, so let's talk about the excretory system. It's several different elements to it here. You see in this diagram several different elements. Begins with the minor calyces, which fuse to form the major calyces, which fuse to form the pelvis, which then continues as the ureter, which then enters into the urinary bladder where the urine is stored, which then releases urine into the urethra to the outside world. Most of these urinary passages are lined by urothelium or transitional epithelium. Here we see a renal cortex. Here we see a renal pyramid. Here we see a papillae of a pyramid. That pointy end is the papillae of a pyramid. It protrudes into a cup-shaped minor calyx, and urine is discharged into the minor calyx. Here we see a higher magnification of the renal papillae, and here you see the largest collecting ducts. Here are the sometimes called papillary collecting ducts, the largest collecting ducts discharging into a minor calyx. On the other side here, the floor here of this minor calyx is lined by urothelium, and here you see the openings of the collecting ducts into the minor calyx. The minor calyces again collect to form major calyces that then discharge into the pelvis, which discharges into the ureter. This is a tubular structure, the ureter. This is a ureter. When the ureter is resting, which it is in this case, the lumen collapses and becomes stellate in its configuration. So here you're looking at the stellate configuration of a resting ureter. If there was urine in the ureter, if it was undergoing peristalsis, pushing urine towards the urinary bladder, the urine would expand the lumen and would look more cylindrical. But now it looks more stellate because it's collapsed. So the epithelium of the mucosa lining the ureter must be able to withstand being collapsed and resting and distended when the urine is traveling through it during peristalsis. So that's urothelium. The layers of the ureter wall consist of mucosa, muscularis, and adventitia. So here, obviously, is the mucosa. This here is muscle, the muscularis. And out here, where you have connective tissue, this is the adventitia. The adventitia, being connective tissue, will bind to adjacent connective tissue, and the ureter will be bound to adjacent structures in that manner. So there are actually several layers to the wall here, which are recognizable if you pay attention to it, and this is just a ureter. These are bundles of smooth muscle out here. That's why they look bundled like this. They're bundles of smooth muscle. This is a urinary bladder. It has also a mucosa, which is lined by transitional epithelium or urothelium. The epithelium sits on a lamina propria. Here you see the muscle layers of the urinary bladder. This is called the detrusor muscle. The detrusor muscle has supposedly 3 layers, internal, middle, and outer, but they are ill-defined layers. They are ill-defined layers. It's somewhat difficult to distinguish the layers, but supposedly there are 3 layers to the detrusor muscle. When the bladder is relaxed, the mucosa gets folded like this. Here you see a relaxed bladder with folded mucosa, and the epithelium lining mucosa is urothelium. Okay. Here's a bladder that's relaxed, and here's a bladder that's distended. When the bladder is relaxed, the wall is thicker, the muscle gets more contracted, I guess, and thicker. When the bladder wall is distended, of course, the lumen gets bigger and wider, more voluminous, but the wall gets thinner. The epithelium also gets thinner. It's hard to see in this figure here, but when the bladder is relaxed, the epithelium is thicker. It might be 6, 7, 8 layers in thickness. When the bladder gets distended, the epithelium gets thinner. It gets to be, let's say, 3 or 4 layers in thickness. You cannot see that easily in this image, but you can see that better in these lower 2 images. In the relaxed bladder, you see there's more layers of epithelium. In the distended bladder, the epithelium gets thinner. There are less numbers of layers. Now, one thing about the relaxed bladder, the most superficial layer of epithelium often will be seen to consist of umbrella cells. These are convex-looking cells like this, sometimes binucleated, like you see in that image. And they're rather wide. They have a larger surface area than the underlying cells, and they may cover several underlying cells at the same time, like an umbrella covering several people at the same time. And as I said to you, they have a convex surface. Here you see how the cells look in a normal, relaxed bladder. Now, when the bladder gets distended, all the cells get more flat. You see they're flattened out. Even the umbrella cells, it's hard to identify the superficial layer of umbrella cells because they become flattened out. But all the number of layers looks thinner. The whole thickness of the epithelium looks thinner, and it's hard to identify the nice convex-looking umbrella cells like we saw in the relaxed bladder. So here, these are both examples of urothelium. And this is what occurs in bladder, and these cells have other specializations which allow the epithelium to not to resist the toxic properties of urine, the hypertonicity of concentrated urine, and some of the materials that may be found, waste products that may be found within the urine, which may be irritating to the underlying tissue. Here's a urethra. The urethra is the last step in the excretory passages of the urinary system. The male urethra is much longer than the female urethra. It's too bad, but it would be nice if there was only one type of epithelium lining the male urethra, but basically, male and female. Also, the urethra is a fibromuscular tube, which is what you're seeing here, lined by up to several types of epithelia. I think it's a bit more complicated in a male than a female, but there may be several types of epithelia. In a relaxed urethra, like you're seeing here, the lumen collapses and becomes stellate looking, like we saw in a ureter, but when it's being used and the urine is coming through, it distends out, becomes more cylindrical in its diameter. But don't be thrown off by the fact that the lumen here is collapsed and rather stellate looking. Is simply a relaxed urethra. So in a male, when you have the part just next to the urinary bladder, in continuity to the urinary bladder, the urethra is initially lined by urothelium, which I don't think is any surprise. At the distal end of the male urethra, where the urethra becomes continuous with the skin on the outside of the urethra, the epithelium transitions to stratified squamous epithelium. So the end of the male urethra is stratified squamous epithelium, and between those two extremities, between the proximal end and the distal end, there are intermediate areas where the urethra is lined by stratified columnar or pseudostratified columnar epithelium. And this diagram from the book actually shows stratified columnar epithelium or pseudostratified columnar epithelium. So that's what's being illustrated here in this particular image. Okay, so it just shows you that the urethra is a bit more complicated than maybe one would expect. And with that, we've come to the end of our review here. Any questions? I'll take them.